Okay, before moving on to the next assay, we have to talk about antibodies. Now, antibodies are proteins, and the shape of an antibody is being shown right here. Typically, the abbreviation for an antibody is just AB, so AB equals the antibody. This is the structure of an antibody, but let's take a little bit more of a closer look at this cartoon drawing, which is mapping out some of the regions on an antibody. An antibody has the shape of a Y, like this. And you can see that you have these constant regions, which are denoted by a C, CH2, CH3, C1, Cl, etc., right? And then you have these variable regions being shown here. Now, these variable regions will be different from antibody to antibody, whereas the constant regions are what is constant between each antibody. Now, furthermore, you can define this bottom area right here as the FC region. That has some importance for us later on. So if I was going to look at my simple Y that I drew here, this top part right here would be the variable regions. And this bottom part right here would be the FC region. Variable, right? Variable. Now, the variable regions, these are the parts that will bind to a specific molecule. Now, what these variable regions bind to are called antigens. So an antibody, through its variable region, will bind to a specific antigen. We'll put a little star here because we're going to say this is going to be very specific interaction. So you get an antibody antigen interaction. Of course, this will be a weak interaction, so let's not put a line here. Let's just put a few dots to say, okay, this is still weak interactions, okay? So we don't get confused later. So where do antibodies come from? Antibodies come from B cells. B cells are a type of immune cell. A very important immune cell. Now, during development, there's a whole library of B cells. So for example, let's say we have 10 B cells. 1, 2. Okay? Now, each one will be producing an antibody with a slightly different variable region. Okay, so we're just going to sort of put an antibody on the bottom for each one. And for example, this one, let's say, has a green circle. And let's say this one has a red circle. And this is just to denote that the variable regions are different. And blue, purple. And now we're going to just change the shape to little triangles instead of circles. Okay, so each one will produce one of these specific antibodies. They won't produce the other antibodies. Okay, so that's very important. And this is because you have, during development, these B cells will go through a process of recombination in a very specific region in their genome where the variable regions are coded, and you get these very unique type of combinations. Now, that being said, each of these antibodies will bind to a specific antigen. So, for example, antibody from this one, so we'll number these so that we can keep track of them. So here we go. The antibody from 2 will bind to a very specific antigen. Since we're going with circles for this one for the variable region, we'll say red circle. Okay? So this will be its antigen. For the number 1, it'll be a green circle. This is its antigen. It could be a protein. It could be a carbohydrate. It could be a very specific lipid, some molecule. Okay? And here we go with the triangles. During development, what will happen is any of these antibodies, if they react with a self-antigen, meaning one of your proteins, then they'll be deleted. They'll be removed from the pool. So let's imagine that this is a self-antigen, this is a self-antigen, and this is a self-antigen. So during development, these will be deleted from the antibody library because this could lead to humongous problems if they're left around, because then you would have autoimmune disease. Okay? So these are removed. We no longer have to worry about them. And through this process of selection, you get a library of B cells that specifically target any possible non-self antigen, whether that's a viral protein, a bacterial protein, etc. Okay? Initially, these B cells are sort of in an inactive state, and we're not going to really go into B cell activation and the immune response, but to get a lot of antibody made, all you have to do is put the antigen into the organism. And that specific B cell will become activated and start producing the antibody versus that antigen. So, for example, if I wanted a lot of antibody for this purple, for this one, for number 5, I want a lot of this antibody, all I have to do is stick this molecule, which this antibody binds to, into an animal, and I will get antibodies produced versus that circle, the specific antibody that binds to that antigen. And then I can isolate the serum from that animal and isolate that antibody from the serum. Okay? So now we have our antibody. Now, why are we talking about antibodies? Because antibodies are a way to detect proteins. And that's what we're going to get to when we're talking about Western blots. Because so far we've talked about how to detect nucleic acid, whether it's DNA or RNA, but we can't use that same technique, a polymerase chain reaction, to detect proteins. But to detect proteins specifically, we need a molecule that specifically binds to that protein, and what better tool to use than antibodies, which are very specific for their targeted protein or antigen. All right, we'll stop here and continue later. Thank you.